they turn on or off, it's somehow like they give the stop or go signal, stop or go sign to the cell. And on the internal and external signal of the around the surround the cell can turn on or off the checkpoint. For example, the internal signal, that is the kinetochore not attached to the spinal microtubule, will send a molecular signal that delays the anaphase. And some external signal, like the growth factors or protein released by the certain cell, that stimulate other cell to divide. For example, the planet-derived growth factor stimulates the division of human fibroblast cell in the culture or even in the body. For example, when you have a cut or a wound on your skin and it the blood come out. So whenever the blood is go out, they can the planets can release the PDGF, the planet drive growth factor, and this growth factor they will come to the um, to the connective tissue under the epithelial cell, the skin, and they bind to the human fibroblast and make a cell signaling to the fibroblast that the fibroblast start to divide and to make more cell. And the fibroblast, if you remember, they can produce extracellular matrix such as the collagen for fiber protein. So by this way they can heal the wounded on your skin. So all the signal come to the fibroblast. It is an external signal. Uh, in this case, it is PDGF, one growth factor released by the other cell. In this case, it's a planet to the fibroblast to induce the cell division. Another example of the external signal is the density-dependent inhibition, in which the crowded cells stop dividing. So most animal cells exhibit the encouraged dependence, in which they must be attached to substrates in order to divide. So in the case of cancer cell, this dependence is defect. So the cancer cell they can divide even there is no space surrounding them. That means even they cannot perform the encouraged on the cell matrix, but the cancer cell they can divide and stay on the other cell, up on the top of the other cell, and make the tumor or the sphere in the in vitro experiment. So that is some example of the stop and go that can control the cell cycle. So the cell cycle control system can arrest the cycle at various checkpoints. In the Z1, the checkpoint can be turned on when the DNA is damaged or there is unfavorable extracellular enrichment. So the normal checkpoint or the R point is the Z1. In the S phase, the checkpoint can be turned on if there is some damage or incompletely replicated DNA. Similar on the Z2 phase, the damaged or incompletely duplicated DNA can turn on the checkpoint. And during the M phase, the checkpoint turn on if the chromosome improperly attached to the mitotic spindle. So the checkpoint is turned off if the cell satisfy all of these things uh, appear in this slide. So when the cell from the Z1 phase in the cell cycle, they want to continue to go to the S phase, they have to satisfy the checkpoint. If they cannot satisfy the checkpoint, they will stop the cell cycle to wait until they have enough thing to um, turn off the checkpoint. But even in the case that the cell cannot satisfy the checkpoint at the Z1 phase, they can dismantle their control system and withdraw from the cell cycle. That means they will go to the Z0 phase and will not divide or repeat the cell cycle. But somehow, even they cannot go to Z0 phase. They cannot go to the S phase. They cannot stay long in the Z1 phase because of some reason, like because of the DMS in the DNA is too large, or the conditions surrounding them, like the nutrient or the space is too full. So this thing can cause the cell to decide to kill themselves, and they call that is apoptosis or cellular program cell death. So apoptosis is one kind of the cell death that the cell can control and can prepare for their death. So it's somehow like they already programmed before, right? So the apoptosis, it is a Greek, the ancient Greek word. So the true meaning of the apoptosis is the how do you say? Is a falling leaf in the autumn. Why they have this word? Because the first scientist who find out the program cell death, he had the question when he looked at the falling leaf from the trees in the autumn. He asked, he wondered why all the leaf falling down from the trees in the autumn, which process inside the cell cause this death. So he concentrated to study on that, and he found out the program cell death or the apoptosis. So he named the apoptosis on this process inside the cell. And it turned out this program cell death is very popular, not only in the plant cell, but also in the animal cell. During the early development and during our lifetime, the apoptosis happens every second, everywhere, and it's like the normal process inside the cell. So here, for example, you see in the image A and B, so this is the finger of the mouse in the early development. So at the early development, you see there is a membrane between each finger, and in at one point, um, all of the cell in this membrane between the finger will be trigger the apoptosis and there and die. After that, the membrane will be degraded and each finger will be separated from each other. So in the age image, they staining the apoptosis marker, and you can see the expression of the marker concentrated on the membrane between the finger as the bright yellow dot. So this is the marker of the apoptosis, for example, like caspase enzyme. So that means all of the cells in the membrane between the finger are trigger the apoptosis, and then they die. So the death of this cell can separate the finger. So after the mouse was born, you see that the finger in the mouse on the mouse um, on the mouse leg or hand, they can separate from each other. And also it's similar for us during the during the early development, our finger still stick together by the membrane development between each finger. And then this membrane will be triggered to apoptosis and disappear. And then our finger can be separate from each other. And you have the normal hand. But imagine that if the apoptosis cannot happen during the development of the embryo, so you will be born with all the finger stick to each other with the membrane between the finger on your hand. So in the lower image, it is the development from the tampon to the frog. So whenever the tampon turn into the frog, so they will lose their tail here. And the losing of the tail, it comes from the programmed cell death of the cell at the junction between the tail and the body of the tampon. So it also the normal process in the development of the frog. And even in our body, right now, in your body, inside the body, many cells are dead from apoptosis. For example, the intestinal cell. So the intestinal cell, they have a very short life with the 12 to 20 hours. So after that, the cell will be triggered apoptosis and they die. So it is very normal process in the body. And of course, the cell can be died from the other way, that they can be died by accident. For example, when your cell contact with the high energy of the light, like X-ray or UV, so they can trigger the cell death, or they can make the cell death. Or if the cell contact with the high temperature, they can be death by accident. So in this death, the cell will cannot prepare for for their death, and um, the the phenomenon of this death is totally different from the apoptosis. So this death they call the necrosis, the death by accident. So look at the image A and B. It is the morphology of apoptosis in B image compared to the necrosis in the A image. So when the cell suffer from the necrosis, you can see that the cell is degraded and the membrane is broken and released on the cytoplasm to the outside, to the environment outside the cell. But for the B case, that means apoptosis, the cell now is under apoptosis. That means they are dying. But you can see that the cell membrane still uh, stay as in compact. That means they still stay as normal, no broken membrane. And inside the cell, the membrane, the nucleus is broken and separate in a small part. And after that, all of the cell will collapse by the cutting of the cytoskeleton, and the cell membrane will wrap around each part of the cell compartment and make an apoptotic body. So by this way, nothing from inside the cell can release to the environment surrounding them. Um, so it like when you how to say, at the end of the day, you always you will put your the waste from your house to the um, to the waste can, right? Something like that. So if you not dividing or classify your waste and put it into different packets and put in the right place. So if you do like that, it's somehow like you the cell do the apoptosis. But if you not do like that, you put all the waste everywhere. So it's somehow like the necrosis, and they can poison the environment surrounding. So inside the cell, in the body, the we prefer the apoptosis than necrosis, because if the cell die of necrosis, they can harm to the tissue and they can poison the tissue, and they can lead to the tissue necrosis also. For example, you see in some wound, if they cannot treat in the good way, so the wound in your body can become necrosis, and the cell will be die, and the necrosis side will develop to the other side of the body. So it is very harmful to the body. And of course, in any way, the apoptosis, if the cell must die, so the apoptosis is much better than necrosis. And in the cancer treatment, uh, the scientists try to find the method can trigger the apoptosis in the cancer cell instead of the necrosis, because if the cancer cell die by necrosis, so the tumor can be release the poison factor to the normal tissue surrounding the tumor cell, the tumor tissue, and they can harmful to the healthy tissue of the patient, and it can um, affect to the life of the patient. So apoptosis is much better because it is a well prepared death of the cell. But of course, we cannot avoid the accident. It called accident because it can happen without our notice, right? So it can happen. But in the case, if you can control, so try to trigger apoptosis instead of the necrosis for the cell. In the C image, it is the one part of the phagocytic cell, such as a macrophage, and they can eat the apoptotic body. That means the one part of the cell death by apoptosis. So each part of the cell can be eaten by the neighbor cell or the macrophage, and they can reuse the cell material in the new cell. So this is very good death of the cell. Here is a video clip illustrating about the cell.
Apoptosis, a form of programmed cell
in
death,
the
has
in
been
vitro
induced in these
experiment.
cultured cells. Cell death is characterized by blebbing of the plasma membrane and fragmentation of the nuclei. Suddenly, cells weaken attachment to the substratum that they have been growing on and shrivel up without lysing. In the following movies, we observe the process at higher magnification.
Okay, so the music is a music from Japanese, because Japanese the samurai, they always kill themselves for the honor, right? So it's somehow like the suicide of the cell, the apoptosis. And this video clip, you see we can trigger the apoptosis of the cell when we can put some drug on the cell culture, and when the cell contact with the drug, they can trigger the apoptosis, and you see each cell will be dead by making some bubble. In fact, this bubble is a apoptotic body, and normally if it happens in the body, this apoptotic will be eat by the other cell. But here, because it is in the cell culture, so you can see the apoptosis stay in the cell medium. Uh, so the apoptosis, they can be induced inside the cell because of the work of one gene family. They call the BCL2 gene family, and as it's involving in the intrinsic pathway of apoptosis. So the intrinsic pathway of apoptosis depend on the mitochondria and involving in the BCL2 protein family. In fact, the BCL2 family, they consist of two type of the protein. One is a apoptotic anti-apoptotic member, and the other belong to the pro-apoptotic member. So interesting, because these two members, even they are in the same family, but they have the contrary function. One member, they can inhibit apoptotic, and the other, they can induce apoptotic in the cell. So they all belong to the same family. They call BCL2 protein family. In the anti-apoptotic member, there are two classes of the protein. Class C1 consists of the BCL2, BCXL, and MCL1, and class 2 consists of the BCLW and A1. For the other, the pro apoptotic member, they consist of three classes, and the class 1 consists of the BAC and MTD BAC. The class 2 is BCLXS, and class 3 is Big Beam, BLK, Bad Bird. I don't know why this name is very funny, but it's on the member of the BCL2 protein for now. They discover and divide into two groups: the anti-apoptotic member and the pro-apoptotic member. So how this BCL gene involved to the intrinsic pathway of apoptosis? So whenever the cell receives some internal signal that the cell should be die, so they can trigger the BCL2 gene to produce the bikes of BAC. Okay, so bikes of BAC belong to the class 1 of pro-apoptotic member. So whenever BAC or BAC is synthesized, they will arrange to make a channel on the membrane, on the outer membrane of the mitochondria. So this channel will release the cytochrome C from the space between the inner and outer membrane of the mitochondria. So cytochrome C is released from mitochondria to the cytoplasm, and in the cytoplasm, this cytochrome C will make a complex with adapter protein and assembly into structure they call the apoptosome. So apoptosome, they consist of the seven of the cytochrome C and other adapter protein complex, and together with seven pro-caspase protein. So the green one is pro-caspase protein, and caspase protein is the apoptotic protein of the cell. So normally the caspase, when they is synthesized, they stay in the pro-caspase state. That means the unmature enzyme. So the pro-caspase will bind to the complex of cytochrome C and adapter protein and make apoptosome. So one apoptosome consists of seven pro-caspase. And when the pro-caspase stay in this apoptosome, they will be activated by the rearrangements of the pro-caspase into the mature caspase. And after the caspase become mature, they will activate the caspase cascade and lead to the apoptosis in the cell. So how is the arrangement or the activation of caspase? So first, the caspase is one family of protease that responsible for the apoptosis inside the cell. They can cut many many type target protein in the cell. And the caspase normally they stay in at the beginning they stay at the pro-caspase, the immature one. After that, the pro-caspase activation can happen by the cleavage of the pro-caspase. And there are two groups of the caspase: the initiator caspase and the executioner caspase. The initiator caspase consists of caspase 2, 8, 9, and 10, and the executioner consists of the caspase 3, 6, and 7. So why is initiator and executioner? The the initiator cascade, they will be activated first, and after activated, the initiator cascade will activate the executioner pathway cascade, and the executioner cascade then will cut the target protein inside the cell and do the apoptosis. So that is why they divide in two groups, the initiator and executioner cascades. Because they all belong to the cascade family, but they also have the two main domain for the cascade. The large catalytic subunits, the yellow, the orange part here, and the small catalytic subunits, the red one. So the two domain, the large and the small catalytic subunit, represent for the cascade family. So all the cascade family have these two domain, and the linker, the domain, linker domain, also the green one. So the executioner, they have three basic type of domain. And for the initiator, they have extra domain like the dash effector domain or cascade recruitment or domain on the initiator one. So this is a pro cascade. And then this pro cascade will rearrange to make the cascade, the major one, and how it arrangement happen. So look at the right image. This illustrated the amino acids peptide, the polypeptide chain of procaspace. So it is the N terminal and it is the C terminal of the protein. In the N terminal, there is one side for the cutting of the procaspase, and the D terminal also. In the C terminal also, there is two sides for cutting. And when this procaspase is arranged in the apoptosome like this, so apoptosome will trigger the cut of the N terminal and the C terminal of the polypeptide. After cutting, the N terminal will be removed and the C terminal will be rearranged between the two procaspase to make the dimer, as you see in this figure. So the two new small catalytic subunits will bind to each other, and each of them will bind to the corresponding large catalytic subunit. So the major cascade will have two small catalytic subunits and two large catalytic subunits. And in this form, they have the activity. They will have the activity. So in the procaspase, they have no activity. But after rearranged with this form, they have the activity of one protease enzyme in the cell. So after the initiator is activated, they can continue to activate the other procaspase belong to the executioner procaspase and make a cascade. That means a, a cascade of activated cascade appear in the cell, and then the executioner cascade will find the target protein and cleave the target protein such as the lamin or the cytosolic protein like the cytoskeleton and the other protein inside the cell. So that is the intrinsic pathway. So in the intrinsic pathway, you see that they involve in the mitochondria and the base or back, that means the BCL2 gene family and cytochrome C. The cell have another apoptosis they call the extrinsic apoptotic pathway. The intrinsic apoptotic pathway involved in mitochondria, as I said. For the extrinsic apoptotic pathway, they not involved into the mitochondria, but they involved to the receptor, on type receptor on the cell membrane, they call the death receptor. And every cell in our body, they have the death receptor. And whenever this receptor bind to their corresponding ligand, they call the death ligand. So they can trigger the apoptosis inside the cell. Okay, so look in the on the left. So when the ligand bind to the death receptor here, they will induce the rearrangement in the cytosolic path of the receptor and recruit the other protein come consists of the initiator caspase called the caspase H. All of these protein come and make a complex called the DAS inducing signaling complex, the DISC. So the that inducing signaling complex will consist of the procaspase 8, the DAD domain of the um, GAD or domain, the adaptive FADD, that means the death domain and that DAD domain and the death domain and the death receptor. So they make the DISC complex, and in this complex, the caspase is the pro-caspase is activated by the rearrangement as similar as a intrinsic pathway that they will be cut the N terminal and the C terminal and we rearrange to make the major one. So after the caspase is activated, the caspase will continue to activate the executioner caspase like caspase 3, 6, and 7. And then this executioner caspase will cause the degrade of the other protein and make the apoptosis inside the cell. So the key of the apoptosis is the activating of caspase enzyme inside the cell. And there is two ways of activating the caspase. One way involving in the mitochondria and the other involved into the death receptor on the cell membrane. So depend on the signals the cell receives, they can trigger either the extrinsic pathway or intrinsic pathway, and both of them lead to the cell death. Um, in our body, the immune cell, like the natural killer cell or the T lymphocyte, they can bind to the death receptor and trigger the apoptosis in the target cell. For example, here you see this is a killer lymphocyte with a fast ligand on their membrane. So when they find out the fat death receptor on the target cell, they will bind to this receptor. And the binding of the ligand and the death receptor will trigger the formation of the DISC complex and activates the caspase protein. After caspase 8 is activated, they will continue to activate the other caspase to make the to make the apoptosis happen inside the cell. And p53 is very important protein for the apoptosis. In the previous slide, I showed you the function of the p53 in the immune checkpoint of the cell cycle, right? If you remember, um, when the DNA is broken and the p53 is synthesized, so they can induce the synthesized p21, and p21 will inhibit the CDK and make the cell cycle stop. So this is one function of the p53. So now you see another function of the p53 in the apoptosis. In fact, the p53 it is the inducer of the apoptosis. They can activate the apoptosis in many ways. Look in this figure. The p53 they can play function as a transcription factor for the synthesis of fast of EGFBP3 and BAX protein. So as you know, the BAX belong to the BCL2 protein, BCL2 family, and BAX belong to the pro-apoptotic member. So p53 they can induce the synthesis of BAX, and after synthesis, BAX can make the channel on the outer membrane of mitochondria and release the cytochrome C and trigger the intrinsic pathway of apoptosis. And p53 can induce the synthesis fast, and the fast is one part of the death receptor, so they can induce the spread of the death receptor on the cell membrane. And the death receptor, if they can bind to the fast ligand, they can trigger the extrinsic apoptosis inside the cell. The third way that p53 can induce apoptosis is that they can um, induce the synthesis of IGFBP3. So, what is the function of the IGFBP3? Normally, inside on the cell, there is a receptor they call the IGF-1 receptor. That means the insulin-like growth factor 1 receptor. So, you can understand in the simple way like this is a receptor for the cell growth or proliferation. Okay, so with this receptor, they have the corresponding ligand called the IGF-1/2, the blue one. So in the normal condition, the IGF-1/2 bind to the receptor and trigger the cell signaling through the PI3 kinase and then the AKT pathway. So this pathway, signaling pathway, they can give the cell survival signal and inhibit all of the protein involving in the apoptosis, such as the BET, the Cas9, the NF-kappa B. Okay, so this protein the involving in the apoptosis and inhibited by the signaling come from the IGF-1 receptor bind to the ligand. So whenever this ligand bind to this receptor, the apoptosis is inhibited. But if the p53 is activated and induce the synthesis of the IGFBP3, so the IGFBP3 they can bind to the IGF-1/2, and this binding will inhibit the IGF-1/2 to link to the receptor here. So if there is no ligand in binding to this receptor, all this signaling is inhibited, and all the protein involving in the apoptosis will be activated. Will turn on activate. That means BET will be activated and Cas9 will be activated and in kappa B also. So the apoptosis will happen. So this is a three way that the p53 can affect to the apoptosis in the cell and induce apoptosis in the cell. Of course, the p53 is sent to synthesize inside the cell if the cell need the expression of this protein. For example, if the DNA is damaged too large and the cell cannot repair, so p53 will be synthesized and induce apoptosis, and the cell will be die. To keep the stable of the cell of the generation, the cell generation. And because of the important function of the p53, so in many types of the cancer cell, they also make a mutant in the gene of p53, or they can make the dysfunction of the p53 protein. So by this way, they can inhibit the apoptosis and can maintain the life of the cancer cell. So p53 also can be used as a marker for the cancer risk on the body. So you see in some, when you come to the hospital, and they can perform some tests, your p53 gene, to see if your p53 gene is mutated or not, because if the gene is mutated, so you have a high risk of the cancer. Because now I think you understand more about the function of p53 in the cancer, because it can inhibit the apoptosis for the cell. And the last part, I want to introduce about the extracellular factor we can control the cell number and the cell size. So the animal cell requires the extracellular signal to divide, and the signal can be classified into the three classes: the mitogens, the growth factors, and the survival factors. So all of these factors, they can together control the division of the animal cell. For the mitogens, how they can stimulate the cell division. So as I already told you about the function of the retinoblastoma, the RP protein. So the RP, they when they are in the active form, they can bind to some transcription factor, the green one. Okay, and the active RB, that means they stay in the hypophosphorylation. That means less phosphorylated. So it is in the normal when the cell stay in the Z1 phase. So the RP protein bind to the transcription factor. But when there is some mitosis appear in the environment surrounding the cell, and this mitosis they bind to their corresponding receptor, the activated mitosin receptor. So they make a cell signaling to the nucleus, and this signaling pathway they can activate the CDK complex with cyclin. That means the Z1 CDK and Z1S CDK is activated. So when this CDK is activated, they will come and find the RP protein and make the phosphorylation on the RP protein. So now RP is hyperphosphorylated and they become inactivated. That means they release the green transcription factor here. So the green transcription factor is released and they can bind to the promoter of the other gene and induce the transcription of the other gene that responsible for the DNA replication. So the cell can do the proliferation. This is the way that the mitosis can affect to the cell division. And for the cell size or cell growth, as I say, after at the end of the M phase, the two daughter cell is quite small and they need time to grow up. So also the extracellular will give the growth factor to stimulate the cell to grow. You see, in our body, the cell have very different in the size. For example, here you have the neuron and lymphocyte. So the lymphocyte is quite small with a diameter of 1 to 2 micrometer. For the neuron, the cell body also small, but the axon and the dendritic is quite long. You can see the axon is quite long. It's about more than 200 micrometer. So very different in the size. So which can control the size of the cell? It belongs to the growth factor and the growth factor receptor on the cell membrane. So if the growth factor bind to their corresponding receptor on the cell membrane, they also make a signaling intracellular signaling pathway to induce the protein synthesis in the body, in the cell, and inhibit the protein degradation in the cell. So the more protein is synthesized, the more size of the cell get. And by this way, they will continue to grow to get the adequate size for the cell division. So it depends on the growth factor and growth receptor spread on the cell surface. The more cell contact to the growth factor, the more size of the cell they can get, and depend on the cell type. Also, the extracellular also provides the survival factor for the cell to maintain their life. If there is no survival factor, the cell can be induced apoptosis. Here is one example of the nerve cell and the target cell, for example, the muscle cell. So on the nerve cell, they have to target to one, at least one muscle cell or the target cell. And in fact, the target cell, they can release some growth, some survival factor surrounding them. And this survival factor, they can bind to the receptor on the synapse of the neuron and then maintain the life of the neuron cell or nerve cell. If there are too much of nerve cell bind to one target cell, so will be not enough the survival factor released from the target cell, and one of the nerve cell will receive less signal of the survival signal, and then they will be induced apoptosis. In fact, the survival factor, when they bind to the corresponding receptor, they also trigger the intracellular signaling pathway to induce the produce of PCL2 protein. And if you remember, PCL2 protein belong to the anti-apoptotic member of the BCL2 family. And when they are synthesized, they can inhibit apoptotics and maintain the survival of the cell. So here is one example of the extracellular growth factor can induce the growth of the cell. Here is a muscle cell. The scientists discovered the mutation of myostatin gene will lead to the dramatic increase in the muscle mass. So when they make the mutant of this myostatin gene, the muscle cells, they can grow without limitation, and they can increase the muscle mass, the huge muscle mass, and you can see the super muscular cow like in this figure. So this is one example of if you understand about the way the cell grow, you can make way to control the growth of the cell in the way that you want. Here another example of the mouse with the mutated myostatin gene and the normal one. So you can see they are totally different in the size, much bigger than the normal one. This is the true. Now it is not involving to the myostatin gene. It involving in the exercise, the sport, and this dog also occupies the mutated myostatin gene. So he developed a very super muscle in his body. Okay, so that is also the last slide for the lesson today, and also the last lesson for our cell biology course in this semester. And I would like to thank you all of you for your participation in this class. And if you have any question, you can ask me now or you can email me. So we are very pleased to respond to all of your comment on your question on your query. And the final exam will be in the next month in January. I think you already received the date of the cell biology exam, and for the final exam is also the multi-choice question. It's also like the midterm exam. Okay, we will take this online, and I wish that all of you can get the good mark on the cell biology course. Do you have any question before we stop?
Dạ có gì thì bọn em sẽ về đọc lại slide rồi bọn em hỏi cô qua email sau ạ.
Ok.
Và bọn em cũng cảm ơn cô vì thời gian qua đã kiểu nhiệt tình chỉ dạy bọn em ạ. Và lớp em cũng chúc cô năm mới thì tràn ngập sức khỏe và càng ngày càng thành công trên con đường sự nghiệp của mình ạ.
Cảm ơn những lời chúc rất là đáng yêu của các bạn. Cô cũng chúc các bạn có một năm mới 2022 rất nhiều thành công và niềm vui. Và quan trọng nhất là cô rất mong các bạn sẽ đạt được điểm cao trong kỳ thi cuối kỳ của môn sinh học tế bào. Rồi, xin chào cả lớp.
Vâng, cảm ơn.